Yeah, happy Valentine's Day, everyone. We're gonna get started as soon as more people are online. If you have a question, could you put it in the chat? Yeah, still have people joining in. I think I will go ahead and get started. Do you think so, Leslie?
Yes.
Okay, great. Yes, good morning, everyone. I want to introduce our speaker, Dr. Brumback, and a good friend of mine, a good friend of the LEND program. And she's a board certified MD, pediatric neurologist, and a PhD neuroscientist at Dell Medical School at the University of Texas at Austin. And her clinical practice and research programs are aligned to understand and help people with neurodevelopmental conditions. She's also been a faculty member in our LEND program since its inception and has been a founding member of the Autism Consortium of Texas, which is where we are now. Um, when we both arrived at UT in 2017. So I'm super happy to hear about Dr. Brumback's work. I'll turn it over.
Fantastic. Well, thanks, Sandy. Dr. Magana, it has been one of the joys of my professional life to get to work with you and get to just see how much you've grown the education and support services for families here in Texas. So I'm going to talk today about autism. I'm going to use autism as kind of a shorthand term for neurodevelopmental conditions. Autism is one component of many neurodevelopmental conditions. And so when I'm talking about the kids I take care of, we often will use a short-term hand, autism. But the people that I'm talking about really today, that I'm working to help in the context of today's talk, are people who have neurodevelopmental disorders that where autism is a component, but there's also associated intellectual disability, developmental delays, cerebral palsy, epilepsy, depression, anxiety, insomnia. And so I just want to make it clear that that's who I'm focusing on today, are the patients who have global neurodevelopmental disorders, and autism is often a component of that. So the journey we're going to take today, first we're going to talk about this, the sense of urgency that I think many of us are feeling when it comes to identifying treatments and potentially cures for these neurodevelopmental disorders. I'm going to talk about that sense of urgency and how that's kind of a new thing for this field. And then I'd like to talk about what are the factors that contribute to our ability to address this sense of urgency from the time a parent has a concern about their child to the time that they receive a diagnosis to the time that we have an understanding of what's going on and potentially a specific treatment. And so we'll talk about challenges with just getting to the point of having a clinical diagnosis, of being told your child has global developmental delay, your child has autism, your child has epilepsy. And then where we go from there and what the challenges and opportunities are from having that clinical diagnosis, then moving forward to understanding why and what we can do to treat that underlying cause. And so that's the focus of today is how do we go from a parent's concern to a treatment for the underlying condition as quickly as possible. So we are in new territory in neurodevelopmental disorders. Right now, there's really no sense of urgency when it comes to getting kids in to see the neurologist. You know, we have waiting lists of 6 months to a year just to get in to see the specialist. That taking 1 year to get in to see a specialist, that is our way of saying as society that this is not urgent, that we're all okay with waiting a year for you to get in and see a specialist. And I think that we used to have this kind of attitude of walking, not running, for other neurological conditions. And so the one that is, I think, the most famous is stroke. And I'd like to make an analogy between where we were in treating and identifying stroke and where we are now, and relate that to where we are right now with neurodevelopmental disorders and make the case that we need to have that same sense of urgency that we do with stroke with neurodevelopmental disorders. So prior to the early 2000s and late 90s, stroke was a diagnosis that could wait till morning. When somebody has a stroke, there's a blood clot that travels into a part of the brain that prevents blood and oxygen from reaching brain tissue. And then that brain tissue starts to get sick and die. And, there used to not really be anything we could do about it. And so the sort of bad old days of neurology was where a neurologist would get a call about a patient having a stroke. And they would say, okay, I'll see them in the morning. There was no sense of urgency because there was nothing we could do about it. And then came tissue plasminogen activator, TPA. TPA is the clot buster. This is a medicine that we can give into somebody's, into their blood system through the vein. And it goes to where the clot is and it helps break it down so that that part of the brain that is lacking oxygen because of that blood clot now can have blood flow again. And so now we have a treatment. Now, when a neurologist gets called about somebody having a stroke, we can no longer say, well, I'll see them in the morning, because now we have something we can do. Now we have a treatment. And prior to the advent of TPA, we were all strolling along, saying, we'll see the patient in the morning, not even realizing that soon everything was going to change. And the field of neurovascular neurology, the field of stroke, would go from being basically something a few people went into to being the most exciting and most rewarding subspecialty within our field. Because you have somebody in front of you, their brain is not working right, we can fix it, and in real time, we can watch them get better. And so now we have an adage, we have this slogan that we talk about called time is brain. And there are, let me see if I can advance my slide. There we go. Now there are public service announcements, warning people, educating people about what the signs of stroke are, so that the person who's having a stroke can have that be recognized, and we can get them that treatment as soon as possible. So we talk about time is brain, every second counts. Stroke, don't wait, be fast. And now it's not about, if you get the treatment, it's how quickly can you get the treatment. It's reducing the door to needle time from the time the person hits the emergency department door to the time we have a needle in their arm, giving them this medicine. I have never felt this kind of urgency with neurodevelopmental disorders. There are a few. Neurometabolic conditions are well-recognized emergency conditions. But for non-metabolic neurodevelopmental disorders, none of us have a sense of urgency. Sure, my wait list is 6 months long. That's, there's no sense of urgency. And so what I'm urging, my sort of collaborators in this space is to take up arms and say, we need to reduce the time from first concern that a parent says to the time that a child is getting a treatment. And I think that we are on the verge of having that be a reality, where a parent's first concern is taken as urgent, that we need to get this child in to see the specialist, they need to get a clinical diagnosis, and then they need to get a diagnosis of the underlying condition so that we can give them a disease-modifying treatment. So I, this past year, for the very first time in my career, finally felt a sense of urgency when it came to a neurodevelopmental condition. And this was because of basic science that was funded through the National Institutes of Health to identify for a neurodevelopmental disorder called Rett syndrome, a disease-modifying treatment. So this child neurologist, Jeff Newell, Alan Percy, Tim Benke, Elizabeth Berry Kravis, and others worked to identify a molecule that is like a molecule that naturally occurs in our body called insulin-like growth factor 1. This is now called trofinetide, this molecule. And after years of basic science, translational studies, painstaking natural history studies by the Rett syndrome community, they were able to then show that there was a clinically significant improvement in patients with Rett syndrome with the treatment of trofinetide. And so I'll show you just what this graph shows. So here on the y-axis is the change from baseline score. So the Rett syndrome community created a Rett syndrome specific a survey and a rating scale for Rett syndrome specific symptoms. And they measured at baseline what that person's score was. And the higher the score, the worse the symptoms. And so as we go negative from their baseline score, we see improvement. And so at week 2, everybody improved. The trofinetide group shown in the circles and the placebo group shown in the squares. And that's the placebo effect. When we look at the placebo group, that initial improvement in symptoms then went away at week 6 and week 12. In the treatment group, the trofinetide treatment group, we see that that initial benefit is sustained through 3 months. And further studies beyond this have demonstrated that this benefit is sustained months later. So this to me is like TPA. This is the clot-busting drug for stroke. This is the version of that for neurodevelopmental conditions. We now have a treatment. And so now, fast forward 6 months, when I met Astrid. Astrid came to my clinic because mom noticed that Astrid was having difficulties starting around the age of
She
9.
almost doesn't even buy it. Like she will buy it,
She
but
just
if you
touches
see
the
it, it's
lip.
just literally right there and repositions.
So what we see Astrid doing is
Oh goodness, that's a good cough.
the standard, the very stereotyped
Tangled on your hand, aren't you?
hand-wringing movements that people with Rett syndrome exhibit. So Rett syndrome is caused by a difference in a gene. And what happens is patients will typically have a typical development, and then they undergo a regression. Their head stops growing, they lose language skills, they lose their ability to use their hands. And that purposeful hand movement is replaced with hand wringing, so non-purposeful hand movements. And so she was referred to me at the age of 18 months. Mom was concerned at 9 months. She got the referral to see me at 18 months for evaluation of gross motor delays and language delays, and a high risk score on an autism screener. So I met her at 22 months of age, and I thought of the clinical trial that I just showed you, and I thought, holy cow, this is Rett syndrome. We have a treatment for this now. And so what normally takes months to get genetic testing approved and the results, we were able to expedite this and get this done in 2 months, to where we went from having her seeing me for the first time to having a molecular diagnosis, having that genetic diagnosis in 2 months. This medicine is approved for people over the age of 2. And so by getting that diagnosis that quickly, we were able to get her started on treatment at the age of 25 months. So time is brain. This was 16 months from the time that mom had her first concerns about her child to the time we got the treatment in her. And so what we need to do now that we're starting to have these sorts of treatments is have this door to needle time mentality that we have for stroke to adapt that for neurodevelopmental disorders so that we can cut this first concerns to treatment time down from 16 months to something much less than that. So there's two ways to do this. One is to get kids in to see the specialists sooner so they can get their clinical diagnoses. And that's what we'll talk about first, is just getting that clinical diagnosis to begin with so that then people can start to think about what might be causing it. And so there are many groups right now who are working on ways to train primary care physicians to make diagnoses of neurodevelopmental conditions, be able to recognize the symptoms of autism and global delays, to be able to make those clinical diagnoses instead of waiting for 6 months to a year to see the specialist. Let's train people who are on the front lines to be able to make those diagnoses with confidence so that we don't have to have that 6 month to a year wait time to see the specialist. So I am going to tell you a few tips for those of you who are clinicians in the audience for how to make an autism diagnosis that might otherwise be missed. So these are tips that I give to primary care doctors, tips that I give to the pediatrics residents and the neurology residents, so that when they see somebody who has developmental differences, they can accurately diagnose that person with a clinical diagnosis so that then we can move forward with understanding why that person has that clinical diagnosis. So the first point is beware litmus tests. So for those of us who did chemistry, there are these little papers called litmus papers that turn blue if the pH is low, is high. So if you have something that's basic, and they turn red if the solution is acidic. And so it was this one or the other. It's either blue or red. It's either acidic or it's basic. There's just two choices. It's a yes or it's a no. And what I'm bringing this up because what I see with clinicians who are interviewing families about a child's symptoms and trying to understand, is this something that is sort of within, typical development, or is this not typical development? I see that clinicians will use litmus tests to say this person doesn't have a difference in development. They'll say this person doesn't have autism because, and then they'll list off things like, well, they make eye contact, so they can't be autistic. They have friends, they have relationships, so they can't be autistic. They do well in school, so they can't be autistic. So know that for many people, experiencing autism is different than the outward appearance of autism. So it's not about whether you can do things, it's about how much energy it takes. And what I find is that people who um, uh, don't feel comfortable um, with autism will say, well, they make eye contact, therefore they can't be autistic, or um, uh, basically just say, because you can do something, it means that we're excluding the diagnosis of autism. And so just remember, it's often about the internal experience and how much energy it takes for you to do things like make eye contact, have friends and relationships, and not about how that looks to you. For adults who have more than one psychiatric diagnosis, I ask my trainees to push the pause button and ask, could this person have undiagnosed autism? What I find is that I have a lot of families where a child has received piecemeal diagnoses of anxiety, sensory processing disorder, social phobia, OCD, and they're receiving these piecemeal psychiatric diagnoses when really the parsimonious diagnosis is that the person's autistic. And so as we are starting to recognize autism more and more in kids, recognizing it more and more in adults so that people who are in adulthood can also have their needs met. One recommendation that I have for people who are specifically understanding if a person has autism is to use a structured interview. And this is one that I particularly like. It's by a psychologist named Donna Henderson and her colleagues, Drs. Wayland and White. In this free PDF that you can download offline, she has a list of things that you can prompt your patients with to ask, do these things come naturally to you or not? Or does this come naturally to your child? And it allows the diagnostic interview to proceed, I think, more smoothly because the person and the family doesn't have to come up with all of these things off the top of their heads. You can prompt them and they can say yes or no, that is or is not part of how I'm feeling. Okay, so we've talked about reducing door to needle time by having the patient go from their first concerns to having a clinical diagnosis. What's next? Once we have a diagnosis of autism spectrum disorder, intellectual disability, epilepsy, what do we do from there? How can we create treatments for people? Well, the first step is really understanding the biology. And so next I'm going to talk about the advances we've had recently in our understanding of the genetics of neurodevelopmental disorders and how we can harness that to develop treatments. So when we look at thousands of people who have diagnoses of autism that are associated with other issues like epilepsy and motor challenges, we find that about 25% of those patients have a difference in the spelling of one or more genes. So if we do a spell check on people, on their genes, and ask, are there any differences in the spelling of these genetic recipes that are important for how the brain works? In about 1 out of 4 patients, we find a difference in spelling. We find a variant in one of these genes that points us in the direction to say, you know what, we think this might be causing what this person has. And so these risk genes fall into a couple of categories. Unfortunately, these categories of function are things that we have a lot of experience with from decades of basic science. So the 2 main categories are, first, genes that regulate other genes. So these are genes that control which genes are expressed. They control whether the DNA is coiled up and therefore not accessible to the proteins that come in to try to read and transcribe those genes. The other class is synaptic communication. So neurons communicate through a synapse. That's the connection between one cell and the other, the presynaptic cell and the postsynaptic cell. And there's a whole host of proteins that are involved in that, both on the presynaptic side and the postsynaptic side. And so most of the currently identified autism genes fall into one of these 2 categories. And so doing genetic testing is not just something we do on a research basis. This is actually part of standard clinical care now. The types of testing that we offer for people with neurodevelopmental disorders are twofold. The first is chromosomal microarray. And so this is a technique where we aren't doing a spell check on the DNA, but rather we're asking, are there any words or sentences within those genetic recipes that have been copied and pasted so that there's a duplication of some information, or a place where a piece has been deleted, where a recipe is missing a component. The other type of testing is called exome sequencing, and this is where we do the spell check, not on the entire genome, but rather on just the recipes that encode proteins. So the recipes that create the proteins that regulate other genes, that create the proteins that regulate how neurons talk to each other. Most insurances now cover these genetic tests. There's also free genetic testing opportunities through research studies, the most famous of which in the US right now is Spark for Autism. And so you register on their portal, they send you a kit to check, to collect a sample of saliva, and then you send that in. If they find a difference in one of these 100 genes that are we call high confidence autism genes or high confidence neurodevelopment genes, they will notify you that they have found this. Then you can bring that to the physician, and then the physician can, on a clinical basis, through your insurance, test for that particular genetic difference, which is cheaper than looking at all of them. Okay, so we have somebody who has a clinical diagnosis. They have a diagnosis of a neurodevelopmental condition. We may, in 25% of people, have a genetic cause for that developmental condition. What can we then do about this now that we have this information? And so I think we're all familiar with the treatments that we have that are for kind of adjacent issues, where we can treat ADHD, we can treat sleep, we can treat constipation, anxiety, we can treat epilepsy. But none of these treatments are really disease modifying, meaning that they're really treating the underlying cause of what's going on. These are what we call symptomatic treatments, where we're not addressing the underlying issue, but we're providing treatment to make the person more comfortable and improve quality of life. We all know about the therapies that we have available from our colleagues in speech therapy, occupational therapy, behavioral therapies, and physical therapies, among others. One of the common therapies is applied behavioral analysis. I often am asked what my thoughts are on ABA, and so I thought I would take a moment to say what my thoughts are about ABA. When parents ask me, should I put my child in ABA? Here's what I tell them. I say, I think of ABA as an extension of parenting, where all families have behaviors that they want to encourage in their children, and they are families are also have a host of behaviors that they're trying to discourage. So we want our children to say please and thank you. We want them to be able to use the toilet. We want them to be able to participate in school so that they can learn. And for many families, the parenting that you do at home is sufficient to mold your child's behavior so that they can function in society. But for many families, they need additional help to achieve that goal of parenting. And so what I talk about with families is, first, when you talk to the behavioral therapist, talk to them and work with them to arrive at a mutually agreed upon set of behaviors that you want to target, so that it really is an extension of parenting rather than you're passing off your child to somebody else to do the parenting that they think is appropriate. So the types of behaviors that I think are optimal and that when I see children with challenges in these domains, I do think it's worth talking to a behavioral therapist to see what they can offer, are really two things. First is, if a person is injuring themselves or others, that is an indication in my mind to talk to a behavioral therapist. And then learning activities of daily living. So being able to use the toilet, being able to sit in circle time, and listen. These activities that we need to do to stay safe, like knowing, to hold hands to cross the street. These sorts of behaviors are essential for somebody to live a safe life. And so those are the ones that really make me say, I think this child would benefit from ABA. But always, the behaviors that are being targeted have to be agreed upon by the caregiver and the therapist. We have educational supports. We have occupational supports for people with neurodevelopmental conditions. And we have social emotional supports. We can help people combat loneliness, who need to find their people. And so we have ways of helping people do that by participating in sports or shared activities of interest with other people. But none of this that I'm talking about is disease modifying. None of these treatments that we've talked about, which are what we currently offer, actually get at the underlying cause. And so I'm going to give an example from daily life of what I mean by underlying cause. For neurodevelopment, I've talked a lot about genetics, but that's quite abstract. And so what would be an example from everybody's daily lives of a clinical diagnosis, an underlying cause, and then the treatments? Well, one we have all experienced is fever. So fever is a clinical diagnosis. We have a high temperature, we have how the person is feeling, they feel crummy. If we wanted to, we can measure levels of cytokines in the bloodstream. But really, this is a clinical diagnosis. And I told you about stroke and how before TPA was invented, we didn't have anything to give people. And so it wasn't an emergency. It was the same thing with fever before we understood what microbes were. So back, before we understood what bacteria and viruses were, and before we had treatments for those things, we would give you aspirin, we would give you Tylenol if you had a fever. And that's great. That helps you feel better. It's a symptomatic treatment. But as long as you still have whatever is causing the fever, you're going to continue to have that fever. And so you can give all the aspirin and Tylenol you want. It's not going to fix the underlying issue. And so as medical science advanced, we became able to say what's causing it. We were able to diagnose different types of fever being caused by meningitis or pneumonia or a blood infection. We were able to understand that there were different ways that a fever could present, different reasons, different underlying causes. But still, for a long time, even though we knew that there were microbes, we didn't have anything to treat to kill off those microbes until the discovery of penicillin. Wow, now we have a treatment. Now, it's not just that we can say your child has a fever and give them Tylenol. We can say why they have a fever. We can say they have strep throat. And now we can actually give you a treatment. And so now when you call your pediatrician's office and you say my child has a fever, that puts into play an entire cascade of diagnostics. And those diagnostics lead to treatments for the underlying cause for the fever. And then your child doesn't need the Tylenol anymore because they no longer have strep throat. And so we're starting to get there with neurodevelopmental disorders. We are at a point now where we can say about 25% of the time what's causing it. We can identify a change in a gene that is important for how the brain develops and functions. And we can say, we think this is, we feel confident this is what's causing it. But we aren't quite there yet with the treatments. And so this is where families are often stuck. You've got your clinical diagnosis of global developmental delay. Now you have a diagnosis for the underlying cause. But what can we do about that? There's unless somebody has done research on that particular gene and put forth a program to develop an understanding of that gene and all the different ways that its function can be changed by these variants, and then set about to find a treatment for that, you're kind of stuck here with just knowing the name of the gene. And so what we're doing at DelMed and across the country are similar efforts, is to take that need for research into our own hands and say, you know what, we could wait for somebody to randomly decide to look at my child's specific genetic difference, study it, develop a treatment for it. But, that's going to take forever. And with over 100 genes and thousands of different ways for each of those genes to be changed, the chances that somebody will study your child's particular variant is essentially nil. And so what we're doing at DelMed is to say, you know what, we're going to take that research into our own hands, and we are going to take our patients' genetic differences, do research on those particular variants, and then find treatments for each person's underlying condition. And so when people talk about precision medicine, that's what we're talking about, is taking a particular person's reason for having an underlying condition, a particular reason for having their condition, understanding it, and treating it at its source. So how do we do that? So fortunately, many of these genes, I would say the majority of these genes that cause neurodevelopmental disorders, are not human specific. These genes are in these two categories, essentially, of genes that regulate other genes and regulating the connections and the communication between neurons. Those are really basic things that we have been doing since we were cousins with yeast, since we were cousins with ants and flies and worms and fish. These gene expression regulation and synaptic communication are things that all animals do. And once we got a good system for doing these things, nature didn't change it. It just kept on using the same machinery. And so the proteins in my cells that are regulating gene expression are the same proteins that are regulating gene expression in ants. And the proteins that are regulating synaptic communication in my neurons are the same ones found in worms. And so now we can say, I don't have to study the person. I can take that person's genetic change and study it in one of these animals. And so we often will use animal models like mice and rats. Those are quite famous model systems to study human conditions. We are using even tinier animal models so that we can do this research quickly and relatively inexpensively. And so I'll tell you about one of the organisms that we're using to take our patients' genetic differences and put them into a model organism to study them. This is a slide from my friend and colleague, John Pierce, who uses this teeny tiny worm called C. elegans. It's less than 1 millimeter long. It only has 300 neurons compared to our billions of neurons in our brain. It only has 8,000 synapses as opposed to the billions that we have in our brain. But the genetic makeup for the way that those neurons and synapses work is essentially the same between the worms and the humans. We have about 16,000 genes that regulate how our neurons do their jobs. Teeny tiny worms have the same number of genes to do those same things. Because in essence, neurons are neurons, whether they're in a worm or if they're in a human brain. And so we now have the opportunity to study these neurodevelopmental conditions that seem human specific, but really aren't. These neurodevelopmental disorders are caused by changes in genes that are for such basic functions, like how neurons talk to each other and how genes get regulated, that we can study these things in these teeny tiny organisms. And so this is our gene to treatment pipeline. Usually a child will receive, and I say autism here, but really I'm speaking about neurodevelopmental disorders in general. And they get genetic testing, and we find the name of the gene. And then that's kind of where it ends, because there's no path forward. There's nothing known about that particular person's genetic variant. There's not enough known about that particular gene. And so usually this is where things stop. We have the name of the gene, but that's about it. What we're doing is modeling that person's genetic difference into tiny organisms like worms and flies and fish and mice, so that we can study that particular patient's genetic variant within a couple of months. And then we can do drug screening. There are thousands of drugs that are FDA approved, but just sitting on shelves because they, didn't make it to market. There's a lot of different versions of Claritin that weren't quite as good as Claritin, or maybe had a worse side effect profile than Claritin does. And so those drugs got approved for human use, but they're just sitting on a shelf, not getting used. We can purchase libraries of these drugs and then ask which one of these drugs works to make the differences that we're seeing in biology better. Which of these drugs treats the underlying condition? And the great thing about this is that we can then just prescribe those medicines because they're already FDA approved. And so what we're proposing is to have a relatively inexpensive, high throughput, and powerful way of going from a genetic diagnosis to a treatment. And here's our proposed timeline. My goal is that this will take 1 year. From the time that we have a gene result to designing and making the animal model, understanding that animal model, phenotyping it so that we understand how that genetic change is influencing its biology, and then to do screening of drugs over a period of 6 months to identify a treatment that we could then prescribe to the child. So I want to close by getting back to this idea that time is brain. We have this sense of urgency for many things in neurology, for many things in medicine, but neurodevelopmental disorders are not one of those things. We are starting to have treatments for these conditions, novel treatments that are becoming FDA approved, but also treatments that are sitting on shelves waiting for us to pluck them off and use them for the right person. And so to go from a situation where we have a 16-month time period from first concerns to treatment, we want to reduce that so that this can happen for everybody, not just the children who happen to have the one or two genetic conditions that have achieved FDA approval for a specific treatment for their condition. So I'll stop there and take questions. Thanks, everybody.
I'm trying to get, and there we go. Yeah, if anybody has questions, we do have 4 in the Q&A, so let's take a look there. I know Benny had this question when you were talking about Rett syndrome, and he's asking what's the oldest a new patient with Rett syndrome can be for the treatment to be effective at treating neurodevelopmental symptoms. Can adults receive any benefit?
So I don't know what the oldest patient on trofinetide is, but I'm pretty sure it's somebody in their 20s. And so this is such an important question of who are the right, who are the first patients that are going to be able to benefit from these types of treatments? So there are, some of these genes cause the brain when it's in utero to develop differently, to where, the brain structure is fundamentally different. Those conditions, I think, are going to be more challenging to treat because many of those genes aren't being used in the person once they're born. Those genes sort of were turned on and they did their thing, and now the person is living with the results of the function of that gene. Conditions like Rett syndrome are pretty common, where the gene, it might be important during development, but it's also functioning throughout life. And so we could potentially change people's biology in real time. Because these genes are operating throughout life, we have the opportunity to help people basically at any age. And of course, time is brain, the sooner the better. But I do believe that even in adults, these types of treatments are going to be helpful.
And I'm wondering, Audrey, like Rett syndrome itself, doesn't it have a short life expectancy for girls who get Rett syndrome? Just talking
That's
about
right.
that specific disorder.
Yeah, so for many of these conditions, there's a shortened life expectancy. And the things that people die of are shared among different neurodevelopmental disorders. For Rett syndrome, they will often have cardiac, electrical cardiac problems. They'll have difficulty with breathing while they're awake, and so they'll stop breathing. People aren't able to protect their airways and keep the food and the liquid going down the right tube, and so it goes into their lungs and they get pneumonias. There's a lot of ways that people with neurodevelopmental diseases can have shortened life expectancy.
Thank you. Yeah, so next question is, if NDDs aren't specific to just humans, why isn't that more widely known? It debunks the age-old vaccine causes autism.
it's a good question. I think that, you know, why isn't this more widely known? I think that this is, the one of the roots of some of the challenges that we're facing right now is that we as scientists and physicians have not done as good a job as others at communicating with people about what it is that we do know. And so, so I think, I mean, that's why I think that, this very webinar is very important so that, we can be educating people about what is known. And I agree, that what we have just discussed is another way to argue against the pseudoscience that's being told to people.
Yeah. Okay. And another person asked, would treating autism with your approach essentially mean they would no longer have autism symptoms present and will develop typically after taking prescribed FDA drugs? Or would it just be lessening of symptoms? I'm assuming this would have to be done at a very early age and it would not be as effective with older patients.
Yeah, I think this is, I mean, I think that first I'll say that, you know, I am a big believer in neurodiversity. And, when I am thinking about the people that I'm trying to help with this, it's people that aren't able to live independently. It's people who aren't able to be left alone for, any length of time because they don't have the capacity to care for themselves and stay safe. And so I do think that it's going to be a case-by-case basis for, how long has it been, the person's been living with this? And what is the particular condition that they have? And if we do find an FDA-approved drug, how good is it? Not every medicine is 100% effective. And so, and not every medicine, and all medicine have off-target effects. And so it's going to be for each person a very detailed discussion and ongoing monitoring to understand, are we seeing benefit? Are we seeing negative side effects? And what's the cost benefit ratio there for whether we think we should continue treatment? I do think, however, that having a basis, having even something that works but doesn't work great is the starting point for developing something that does work better. And so that's where people who are doing drug discovery research will come in, that we can go to them and say, I found, this molecule that seems to help a little bit, can you modify it in 1,000 different ways? And then we can test which one of those is a better version of what it is that they're taking. And then we can test that new medicine to see if it improves symptoms even more.
Yeah. And I'm wondering from some of these questions whether people are thinking that there are FDA approved drugs for any neurodiverse. I feel like what I heard you say is that there is a drug that was established for Rett syndrome, and that going forward, the future research is going to look into, but there's there's not a series of approved FDA drugs that are covered by insurance that to treat autism at this point or other things. Can you clarify that?
Yeah, so at this point, there's less than, I would say less than a dozen. And they are for different conditions. Some of them are gene therapies. So there's a condition called spinal muscular atrophy where the neurons that are in the spinal cord that innervate your muscles die off because they don't have this gene. We now have a treatment that if we can detect it early and we can provide the treatment, then those motor neurons survive and the person doesn't have the symptoms. And so that's like a miracle. You know, these are kids that used to die because they weren't able to breathe. And so if you were born with the most severe version of this condition, you wouldn't live past the age of 1. Now these kids are going to school and playing on the playground. So there are real examples of, you know, cures for conditions that are neurodevelopmental conditions.
Are they generally rare genetic disorders that you're speaking of, or?
Yes. And so that's where I think it's important for me to say that neurodevelopmental disorders, they're really common. Like there's, you know, we think, you know, maybe 20% of people have, you know, some kind of neurodevelopmental disability if you include everything from learning disabilities to intellectual disability. But each one of those people is unique. And each one of those people has a unique reason for why they have that clinical diagnosis of a neurodevelopmental disability. And so, so maybe I'll use the example of fever. So we have, once we discovered penicillin, we had treatment for a specific bacteria that caused specific types of infections. We had not yet discovered antibiotics that treated other types of infections. And so this was, you know, but we had to start somewhere. We had to start with, you know, individuals who had that particular, you know, an infection caused by that particular bacteria. And for those people, it really worked. And so that's, you know, how this is going to go with neurodevelopmental conditions is we have to understand why this person has their neurodevelopmental condition. What is the underlying cause for you? And a treatment that is designed for you isn't necessarily going to help, you know, thousands of other people. It might just help you. So I do think it's important, you know, while I think that we as a field are rapidly moving towards having targeted treatments. What we're trying to do at DelMed is really build the structure, build the infrastructure so that we can get genetic testing on every child. So we can then do precision medicine on every child. We are not there yet, but that is why we now have a Center for Rare Disease that is going to, you know, be a part of our gene to treatment program, is going to be, you know, housed within the Center for Rare Disease so that, you know, we're all working together towards the same goal of being able to provide precision medicine to everybody.
That's awesome. And maybe in that context, does insurance cover this?
Yeah. Oh, insurance. You know, so the medicine for SMA, this muscular disorder, this neuromuscular disorder, I don't remember what the current cost is, but it's something like $500,000. You know, it is expensive. And so there's been a lot of, you know, ethical debates about the cost of a one-time treatment versus the cost of, you know, a lifetime of, you know, the supports that that you would need if you didn't have that treatment. And so it's going to be a very interesting time. And I think that patient advocacy is going to be really important to demonstrate to our elected officials that the benefits of having treatments that are coming out is something that should be covered by insurance, or that we should have some sort of structure to our medical system that allows people to access these treatments.
Yeah, thank you. And Angela says, thank you for speaking. Thank you very much for speaking on ABA therapy. I always appreciate the discussion. As a parent of an autistic child and an adult who is learning to accept some of my own differences, it has become such a heated topic in some spaces. I just want to do what's best for my son. So not a question, but gratitude for you and your work.
Yeah, thanks. I think, you know, uh, when when I sort of when I think about uh, any type of therapy, it really comes down to the person. It comes down to the therapist and their relationship with the family, their relationship with the child, what the shared goals are. And I tell families, who are, gosh, I don't know if I really, want to do this. I say, go talk to them. Go talk to, the people at your local center and find out, like, are these people you want hanging out with your child? We all do this when it comes to, picking basically anything for our families. Is what does our spidey sense say? Sure, we can talk about sort of the, the idea of these things, but when it comes to, educational settings for your child and, what we should be asking for with the IEP, to what types of therapies and how frequently, and, it really is all about your spidey sense and what you feel like, your child needs, and whether the people that you're interfacing with understand that.
And then Mr. Gregory or Mr. and Miss Gregory asks, any developmental treatments for symptoms from mice to gene mutation?
Oh, I will have to look that one up. I don't know about that one. So I think that for the, people doing clinical work in the room, I will say that, it used to be that we had, it used to be that there were like 5 antibiotics and, one cancer treatment and, 5 neurodevelopmental syndromes that we recognize. Now there's 1,000 antibiotics, there's 1,000 cancer treatments, there's 1,000 different genes that we now know cause neurodevelopmental conditions. And so there's no reasonable expectation that you should know about every single one of those. And this is where this is where having I think the family foundations that are established for different genetic conditions has been so helpful for the medical community who serves families, is to have a go-to source for information about that particular condition. So when I have a patient with Down syndrome, I go to the Down Syndrome Foundation website and I download the PDF of here are all the things that you need to be thinking about in a patient visit. And they have all of the research in one spot that has been done on that particular condition. And so it's really about knowing where to look for the information. And so when I get asked, what about this particular gene? Like, I don't know, but I know where to look. And I know how to find whether that is something that is already known or not. And so I think just sort of for clinicians, especially early in your training, it can be hard to admit that you don't know something because you feel like, I don't know, what's reasonable for, I should know everything, right? But I think that as you move along in your training, you find that, yeah, there is so much to know, and you learn basic foundational principles, and then you keep up to date with the details as you go. And in the neurodevelopmental space, that includes, learning about each of these different genetic syndromes.
Okay, and Shuning Huang asks, for ASD with known specific genetic mutation, how will your approach work? Will it still follow the timeline shown in the slide?
Yes. So right now, we are putting together from patients in my clinic, I'm compiling a list of what genetic conditions my patients have and whether what they have is something that we would be able to do right now through this gene to treatment program. And so we have a handful of families right now that are in this pipeline. Our very first patient, we have now completed the first half of this, where we create the animal model, we identify what are the biological differences. And so now we're ready to move towards doing the drug screen. And so, so I think that as we're in this startup phase, you know, working with the community to, you know, figure out ways to really scale this up, so that we can be serving more people. Because right now we're a plucky skeleton crew, and you know, to really do this, to really be able to bring about that, you know, first concerns to treatment, you know, mindset of urgency. You know, we need to be able to scale this up so that we can be treating our entire community.
Yeah, thank you. And Leonid asked, I believe some advocacy work to encourage the FDA to expedite your proposal would be beneficial. Is there a petition? How can I help as a clinician?
I think that right now, my recommendation for everyone, every person, is to call your senators, call your US representatives, and call the local office. I called Lloyd Doggett the other day, and a human picked up the phone after one ring, and I got to talk to this person for 10 minutes and tell them all the things that I'm concerned about. And I asked this person, like, does this help? And he said, absolutely. There are so many things going on. We need to hear from people about what they want us to focus on. And so I think that this is something that we can all do, is get on the phone, call these offices. I didn't have anything prepared. I just kind of rambled for like 7 minutes about everything that came to mind. And, you know, it was fine. I was expressing myself, and there was a desire to listen. And so I think that that is the very first thing we should all do, is just call our elected officials and say, you know what, in order to, you know, provide these kinds of treatments, you know, we need to support the basic science so that we know enough about these genes to be able to design a treatment. And so, and, you know, we need to be able to, you know, have the FDA, you know, really work with disease advocacy organizations to move things forward, because time is brain. And so I don't have a specific, you know, petition or anything, but I think just speaking to the people who are supposed to be representing us about this and making it clear how you feel.
Absolutely. And along those lines, T. Gregory said, how can families, individuals get involved with your mission of testing for precision medicine?
So I guess I think that the way to do this would be to send me an email, and I'll put my email here. And I will just say I am one person, and I just want to set expectations because, you know, my goal is to help every single person. What I'm trying to do right now is figure out how we can build this, and bring on more team members to be able to make this so that, you know, we can offer this. And so please be patient with me. And, if I don't get back to you, please forgive me. I'm doing the best I can to try to spend my time, moving this forward. And if I respond to every email, I'll just be sitting in front of my computer responding to emails all day. And so please forgive me. I'm not trying to be whatever, you guys understand. So anyway, but please just email me and let's, be in touch. Yeah.
Yeah, great. What a great set of questions too. And thank you for your thoughtful responses. I know one person on the chat said, thank you for this informative presentation and explaining it with your personal thoughts. So your style is just really resonating with people, I think, in terms of, breaking it down and from your perspective. So that's so great. So I think that's all we have for today. Did you want to say something to the group, Leslie?
Yes, we'll be sending out, you'll receive a survey, and if you could please complete that, that would be wonderful. And then I will be sending your CEUs, but there's a lot of you here today, so give me until next Friday, but you should receive it by then.
And
Awesome.
please, when you fill out your survey, if there's a place to put comments and feedback for me, I'm a work in progress. I'm trying to, figure out the best way to talk about these really, complex issues. And I want to be doing this well. And so please give me your feedback because I really, I want to be doing this right.
Yeah, and there is place, right, Leslie, on the survey form for people to fill in
Yeah, thank you.
their
I
thoughts
really
and
appreciate it.
comments. That'll be great. All right, well, thank you so much, Dr. Brumback. That was fantastic. And as some people have said here, transformative presentation. So we really appreciate that.
My
Yes,
pleasure.
thank you.
Yeah, thank you to everybody. And thank you for everybody here is doing work in this space. And so we are all in this together. And so thank you to every single person here who has taken the time to be here and, participate in this work that we're all doing.
Yeah. All right. Thank you, everybody.
Bye.